Thank you, Routine, for the introduction. Good morning, everybody. I'm Julie Thomas. I'm a faculty member at RIT in the School of Life Sciences. I'd like to thank you all for listening to me today and having the opportunity to present as part of this community. I confess I feel got a little bit of imposter syndrome because I really have done very little hands-on EM, dating back to when I did it as a grad student, characterizing phages I'd isolated on what was then a very old electron microscope. But at that point, I kind of learned how important EM is for characterizing phages and developed a huge respect for people that actually do EM every day. So before I talk a little bit about this phage SBN3US that we are working with, I thought I'd just give a quick shout out to all the people that have helped make this research happen over the years. I've been very fortunate to have some fabulous collaborators, and I'll be showing results from mass spectrometry that was done by Susan Weintraub, who's at UT Health in San Antonio. There'll be some beautiful images from Ru Ching. And I won't have time to talk about today the collaborative research that's going on between my lab and that of Blanca Lepisco-Enchanas, who's in biomedical engineering at RIT, but they're trying to develop novel ways of purifying phages using electrokinetics. And of course, I've got to say a great big thank you to the current and former members of my laboratory. At RIT, we are primarily an undergraduate institution, so I teach, and I also have a research lab, and I have a lot of undergraduate students, including here Carmen and Fletcher, who are currently in the lab. Today, there are numbers of students who have what I'm presenting today, there'll be numbers of students that have now graduated, whose work has also really been important for what I'm showing today, especially Aaron Schoig's research. So I thought I'd start just giving a brief introduction about what tailed phages are, why we care about them, and then introduce these kind of large, odd jumbo phages, and kind of why we're interested in their capsids and how they assemble, and kind of this process of proteolytic maturation that we kind of know is now there's getting to be some more sense to this process that occurs during head assembly. So tailed phages are basically they all have an icosahedral symmetry-based capsid that's formed from this very thin, strong outer shell formed from the major capsid protein. At one of the vertices of the capsid is a specialized ring, the portal ring, that is multifunctional, has roles in assembly, DNA packaging into the capsid, packaging of that double-stranded DNA genome, and also then attachment of the tail. And these there are three proteins, this major capsid protein, the portal, and another protein that's not part of the mature virion, the terminase protein, that are conserved. Those folds in those proteins are conserved in all tailed phages. They're very anciently derived. Tailed phages come in different flavors of morphologies, and they can be categorized into three main tail morphotypes: these short-tailed podoviruses, these long flexible-tailed cyphoviruses, and then these phages with these much more complex tail structures, these contractile tails, that myoviruses have. And the phages I'm going to be talking about today primarily fall into this category. So The tails of tailed phages are really important because they actually recognize the bacterial host cell, um, and basically they recognize a receptor on that host cell, and there is ejection of the phage genome then into the host cell. So tailed phages can have two kind of main decisions, life cycles, after they eject their DNA into the host cell. Some phages, that DNA, their genome is integrated into the bacterial chromosome, and that's a beautiful long-term mechanism of maintenance of that phage genome that's then called a prophage during replication of the cell. But the phages I'm going to be talking about today are what we call lytic phages. They primarily can only go through this lytic life cycle that has a productive infection. So they commandeer all the resources in the host cell to replicate new viral progeny, and those progeny are released at the end of infection by this process called lysis that is driven by phage-encoded proteins and an enzyme, an endolysin. And this is very, very dramatic process. You can see here we've got a salmonella cell that is basically just disintegrating about 30-40 minutes after infection by this phage SBN3 I'm going to be talking a little bit more about. So it's this lysis that's really important for applications of phages, this killing of the bacterial cell, and probably the most well-known application of tailed phages, for which there's a great interest right now, is phage therapy. That's using tailed phages to kill pathogenic organisms. It's a very it's been used in some countries for a very long period of time, such as Georgia and Poland. And there's a lot of interest in it just globally now because of this problem of multi-antibiotic resistant bacteria. And so there's now even centers in the US, such as at UC San Diego, the Pittsburgh phage program, Yale, and Baylor. They're all having these centers where people can actually get phage therapy on a compassionate use basis. And there's advantages to phage therapy, just like with antibiotics, and there's also disadvantages. And we don't do phage therapy in my lab, but what we're kind of trying to do is do research on a phage that's relevant for phage therapy to kind of start to pull apart this problem of genetic dark matter in tailed phages. And that means basically tailed phages represent the most genetically diverse group of entities on the planet. And for many of them, we probably only know like maybe 70, 80, we probably only know functions for 20 to 30% of the genes that their genomes encode. So if we don't know what their genes are doing, we don't really know how best to exploit them for biotechnological tools. And my lab is really primarily interested in these really complex phages that have long genomes. And so this problem of genetic dark matter is kind of like, it's amplified because they have genomes that are over 200 kilobase pairs long and hundreds of genes, a large proportion of which are functionally unassigned. And the prototype of the jumbo phages, and for which now there are hundreds of related phages have been isolated, is Pseudomonas aeruginosa phage, PHKZ. You can see here in this micrograph, it's a myovirus, it's got a contractile tail, and they also included in this image variants of T4 phage just for comparison purposes. These phages, these jumbo phages, are really odd. They have many virion proteins, upwards of 70 or more different proteins forming their virions, and usually about 50 of those proteins are forming the capsid. And what's really odd is that not only do they have DNA inside the capsid, they also have this great big bolus of proteins, which are thought to then be ejected into the host cell at the start of infection. And this was first discovered by Viktor Krylov over 40 years ago now, and we still know very little about it. But I've been curious about these internal head proteins of these jumbo phages for a long time, and how do they assemble these capsids? So Alistair Stevens' lab did a beautiful reconstruction of the phage head, and this is now, would be not considered a high resolution reconstruction, but it was really giving us a lot more information about this bolus of proteins inside the phage head. And you can see, like, within the head that those proteins are kind of represented by these bubbles, which you start to see when the virions or the heads are exposed to extended doses of the electron beam. And so, what is the function of this in a body? Is it have just roles in host takeover? Does it have roles in stabilizing such a large capsid? Because the capsid has a T = 27 triangulation number. Or does it have a role in assembly? About that time, I was in Lindsay Black's laboratory, and we were also doing analyses on the composition of the phage head. And we identified, using mass spectrometry, that there are about 50 different proteins in the phage head, and that 19 of these, there was evidence that these proteins had been cleaved after a glutamate residue, after this very short kind of sequence motif. And we also saw that these cleavages were kind of like messy, and that for some proteins, we identified there'd been multiple cleavages in them, which didn't really make a lot of sense at that particular point. We identified that there were numbers of proteins that were really high abundance, such as GP93 and 95 and GP162, that we estimated were present in the head in at least 100 or more copies per head. And we also identified the protease responsible for this proteolysis of the head proteins, and found that it was also cleaved. It cleaved a propeptide from its C-terminus, whereas all its other substrates, there was N-terminal propeptides. And we found that there was diverged homology between the phage protease with that of T4 phage. Which was really exciting. We saw this similarity between the proteases and also this cleavage motif, because in T4, the T4 head has head proteins, 9 head proteins that are cleaved after this. They cleave C-terminal to a glutamate residue after this very short sequence motif. So just to quickly go through this was important because we could kind of then try and infer what we knew about T4 to learn more about phage, although the two phages are very, very different. In T4, head assembly starts at a portal ring, and then there is this assembly of kind of all these other internal head proteins that help determine the size of the head. Some are also ejected into the host cell, and then there is this outer shell kind of assembles around it. At that point, something triggers the T4 prohead protease to activate. It cleaves a whole lot of short propeptides from the N-terminus of the internal head proteins, making space inside the capsid, as well as the expansion of the capsid to an icosahedral kind of prolate form, so that the genome can then actually be packaged into the capsid. So this proteolysis step was known to be essential in T4. If proteolysis was prevented, such as in a protease mutant, the proheads would remain anchored to the inner host cell membrane. And all these steps, a lot of them, what is known from the beautiful genetic work that was done with T4. So kind of knowing this was really a big incentive for me to kind of move to, I'm so sorry, for some reason it's not moving forward. There we go. To move to this phage that infects Salmonella, SBN3OS, which is related to VKZ. It shares a core set of genes with VKZ, has a lot of structural similarities. It has a T equals 27 capsid, has a bolus of proteins within the capsid, many of which are homologous to those in VKZ. But one of the driving incentives to work with SBN3OS was because it works, because it infects Salmonella, for which it's a genetically malleable host. So we've done a lot of work with SBN3OS. We've done a genetic analysis. We've done a lot of electron microscopy of mutants over the years, and we've also done a lot of proteomic analysis and really been trying to, identify which proteins are cleaved that form the head and where they're cleaved. So we know that we have within the SBN3OS capsid, there are 9 proteins that undergo proteolysis. They have an N-terminal propeptide that's removed. We know that the cleavage of all these proteins occurs C-terminal to a glutamate residue, just as in, we've seen in T4 very diverged phage, and in VKZ very similar motif. And, we know from this evidence that we see in the mature virion that there has to be a massive remodeling of the head as it goes from this prohead form to the mature form. And even if we just sum up, the amounts, just estimate the amounts of protein that are removed from the head, looking at the propeptides of the major capsid protein and the 2 very highly abundant paralogs, GP53 and GP4, that we believe form the majority of the internal head protein. If we sum up all their propeptides that are removed, that equates to over 40 megadaltons of protein that's actually leaving the head. But we say leaving the head, but we've long known that there is evidence that leaving the head of the propeptides is kind of messy. We know the propeptides are cleaved in multiple places, and we presume that's to make the propeptides shorter so they can exit the capsids. But the fact that we can see that we've been able to find these internal cleavage sites of the propeptides has always kind of suggested to us it's a messy process. We do know that the majority of each of, the proteins that are in the capsid that are cleaved, they are cleaved at a maturation cleavage site though. And this is just showing you, in one of the gel LCMS gels that we've, had analyzed, the gel lane is, cut into many slices to separate out the major kind of components in each gel band. And you can see here, this is the sequence coverage in yellow and green for these internal head proteins that are highly abundant. So we see that the N-terminal propeptide is completely removed in those bands. But just to kind of give you some evidence that we know that this process of this removal of those propeptides is kind of messy, if we see this summary of where we're identifying in the gel propeptides, and again, these are our highly abundant proteins, so we'll see some peptides for them in various lanes of the gel. But importantly, we have identified that there are peptides from the propeptide regions in various lanes as well, which we always thought was kind of evidence of kind of just that the proteolytic maturation going from that spherical to that expanded form was really fast. The protease cleaved rapidly, and there just wasn't time for those propeptides regions to be completely cleared from the internal part of the capsid. What has also been really nice working with SBN3US and doing genetics is that we can kind of start to break genes, see what goes wrong when we knock them out. And we're really fortunate that we've got multiple protease mutants. And so we've been able to study these mutants, such as AMBER59, and you'll grow them under non-permissive conditions where that protein is not expressed. And you can see, looking at the sequence coverage of the major capsid protein, there is the N-terminal propeptide. We see that sequence. There is no cleavage of any of the normally cleaved head proteins in this particular mutant compared to like in the wild type phage. And you can see we kind of get these, they kind of beat up particles, but they're spherical. There is no DNA in them. And there's obviously not, the tails are not able to attach to them either because they haven't gone through proteolytic maturation and DNA packaging. What was really also beautiful was using these mutants. We've seen that the proheads do actually form on the inner membrane, just like in T4, even though these two phages are very different. This is a very apparently a very anciently derived mechanism in head assembly, and that if you don't have proteolytic maturation, your proheads will just stay remaining on the inner salmonella membrane, even after the cell goes through lysis, which is kind of delayed in this mutant. So obviously you don't produce any viable progeny from such infections. So this again, as I said, linked us back to T4, which is really, really beautiful. Seeing this similarity of when you have a protease mutant, your proheads are still, they cannot go through proteolytic maturation, and the proheads remain anchored to that inner membrane. Also, what was really beautiful was Alistair Stevens' group did analyses of these particles from the protease mutant AM59. And if you see here, we just see these images of the mature capsid compared to these proheads from this so-called modeled capsid derived from AM59. And you can see that there is kind of like this inner ring that has a T=1 symmetry. And we know that your internal kind of shell of proteins is composed primarily of those paralogs GP53, 54, which are divergent homologs to the PHZ internal head proteins. So we believe that these proteins have a role potentially in head assembly, but then they also have a role in head being ejected into the host cell. So these are these proteins prior to proteolytic maturation. What was really beautiful as well is it kind of like it was Bernard Heyman that did these reconstructions, was showing that there were interactions between the outer shell and that kind of that inner shell as well, formed by that protease mutant. And I'm nearly there. Um, and what's really, really exciting is it's kind of like there's evidence coming from different angles coming to show that these internal head proteins that are highly abundant in these jumbo phages, they are assembled kind of underneath, the capsid shell, or the capsid shell is actually assembled onto them, and that actually after proteolytic maturation into the mature capsid in phage, it was shown by this group, Young et al., that there's actually the propeptide regions have this role in kind of helping stabilize that mature T equals 27 capsid. So you can see here, these are the propeptide regions of GP93, that highly abundant internal head protein of phage. So basically, we still have a lot to go to learn about with jumbo phages. We know they have these very highly complex heads. We know that proteolytic maturation is this really essential process, but we really don't still, there's so much about what drives it, what activates the protease itself, to learn and to better understand where each of these head proteins are within the capsid itself. And with that, I'd like to thank you all for your attention. Thank you.